13 minutes. I don't know why that's happening. If any of you are like YouTubers and use the gamey thing, and you can explain it to me, that'd be great. If you're not, maybe I'll ask ChatGPT if it happens again soon. Okay, you can't see it, I can see it. The problem is that when it goes away while I can see it, while I'm lecturing it, I sometimes don't catch it.
Can you make it so that we can see it like last time, so we can let you know?
I don't know.
Okay.
That would be great, wouldn't it? But anyway, I can work on that as well if it ends up being a problem. Okay, so we got a quiz next week. We don't have class on Monday, right? So we have lab next week. There's not going to be any pre-lab with that. But we'll go ahead and we'll start lab notebook quizzes next week. So be ready for one of those at the end. Okay, so we're going to start writing things down again. What else do we have going? I think that's it. You guys think of anything? We'll have the usual help session on Thursday, 8:30 to 9:30 p.m. But if nobody's there at some point during if we get to 8:40 and nobody's there or later, I'm gonna log off and I'm gonna start drinking. So that's when our help session is, and then remember we've got Monday, Friday, office hours, 1:15 to 3:15 p.m. and Friday, so I've got office hours today, 1:15 to 2:15 p.m. and outside of that by appointment. We'll try and figure out times that work. Okay. Questions? All right, then let's go on and we're going to talk about, what I want to do is I want to talk a little bit about things that happen at the end of glycolysis. All right, so at the end of glycolysis, we get a pyruvate. So for now, what we'll do is we start with glucose. We have glycolysis. And then we get pyruvate. And we talked last semester, we made a big deal about it because we actually had a lab that was related to it, about how if you don't have enough oxygen around, you might do something else with that pyruvate to regenerate the NADH, right? You've got this NADH. Actually, I should say we were generating the NAD+. We're making NADH. So if you're a mammal, you're an animal, what do you do with that pyruvate to regenerate NAD+? You make lactate, right? So what you're going to do is you're going to take lactate dehydrogenase, and you're going to use that to make lactate. I'm going to go ahead and take our pyruvate, and let's go ahead and let's look at the molecules here, because we can see, that way we'll be able to see oxidation and reduction. So there's lactate. And then if we're going to, I'm sorry, that's not lactate, that's pyruvate. And so if we make lactate, what we're going to have is we're going to reduce that. Okay, and we're going to reduce that. In the process of reducing that, what we're going to do is we're going to take this NADH right here, and we're going to make it into NAD+. So we're going to use that NADH to make the lactate, but the whole point here is we're regenerating NAD+ so that we can run glycolysis a little bit more. Okay, we're not getting a lot of extra ATP this way, but apparently it really matters because every animal has lactate dehydrogenase and it has a lot of it, which is why it's a great thing for that lab. Okay, however, what do you do if you're yeast? You're gonna do the same sort of, we call it fermentation, but you're actually making ethanol instead, which is great for brewing. And so you have an enzyme called pyruvate decarboxylase. What do you think it does?
Carboxyl.
It decarboxylates. It takes off a carboxyl, right? Or carbon dioxide. Okay. Very often these enzymes are named for what they do. So when you're trying to memorize glycolysis, that will be helpful for some of them, right? Unfortunately, for sometimes they got named for the reverse reaction than the one we normally talk about. All right. So this lactate dehydrogenase would be something that takes a hydrogen off the lactate, which this does. I can take lactate and I can put NAD+. You did this last semester. I can make NADH out of it. But that's not the way we usually talk about it, right? It does happen. So the reverse reaction happens in the liver. So pyruvate decarboxylase, we're going to lose a carbon dioxide, and that's going to give us an acetaldehyde. So we end up with that. Okay. And then we have alcohol dehydrogenase. And that's going to go ahead and make ethanol. Okay. And this, what am I not drawing here? What's the whole point? What did I forget? Right, we need to make NAD+. I'm going to pull it over as a red arrow, I need NADH, just like over here where I had the carboxylate, right? Right here I have the carbonyl, and I reduced it to make an alcohol. Right over here I've got a carbonyl, and I'm going to reduce it to make an alcohol. All right, so microorganisms do that instead. Generally, we refer to both as fermentation, at least in terms of from a biochemical perspective. It feels a little bit weird to talk about fermentation happening in your body, but it does get called that, get named that way. Questions on that? Yes?
Is there like a disease or something where you do fermentation instead of like...
Are there ways that you do it, personal, animals do it? No.
There's that one condition where you basically can like, you can like brew like alcohol in your, because of the microbiome in your gut, you can have a specific
That
condition.
it can do that.
Yeah.
I've never, find the name for that. I want to look that up. What is it called? Autobrewery syndrome. I'm going to write that right here. Yeah.
The funny part about it is just the fact that you expect that your breath to have way more alcohol in it than it should. You will fail breathalyzer tests comically. Like,
Interesting.
50% blood alcohol content is what it reads because you just exhale the alcohol.
Right. Interesting. Well, I'll have to look that one up. Yeah, it's not. I was gonna say that's related to diabetes, but there you're actually breathing out acetone. We'll talk about that later. But okay, I had never heard about that. That'll be interesting to read about. But this is what bacteria would do that. You do this in terms of, but the whole point. This is a way that pyruvate can be used. Okay, besides taking pyruvate down into the citric acid cycle. All right. So I'm going to move on, though, from talking about pyruvate, and we're going to talk about ways that glucose can be used, or more specifically, talk about glucose 6-phosphate, right? We talked about how we have glucose 6-phosphate. And once we have it, you can go ahead and you can turn it into pyruvate through glycolysis. Alternatively, what else can happen? You can do the pentose phosphate pathway, right? Pentose phosphate pathway, and that's going to give us ribose and NADPH. The ribose is good for making nucleotides, right? Making DNA, and the NADPH is good for recharging an antioxidant system, and I'll take that as fine. It's also used for synthesis, right? So fatty acid synthesis, some other forms of synthesis, you're going to use that as your energy source. And then the final thing that we've talked about, which is what I'm going to go talk about today, is what's the other one? We can make glycogen. So if we want to store that glucose. Sophia, did you have a question?
Yeah. Can you just like say what the difference between NADPH and NADH?
NADPH, the difference between NADPH and NADH is a phosphate. And that's it. So if we go back to, I can't draw these structures from scratch, although I can draw them. I can do what I've asked you to do, but I can't.
I know that's the phosphate sense, but like, why would you use one or the other? Like, why is NADPH here instead of like?
Right. Why? Because they actually are about the same energy. It's about the same. Sometimes one enzyme can use the other, and it's about saying this right here. This is for making ATP, as for on. I put a phosphate here. This is used for other things in the cell. And I don't know. I don't have a good explanation for what the reasoning is, but it may be that this helps compartmentalization. Fatty acid synthesis occurs in the cytosol, and so maybe you put the phosphate on, that means that this NADPH is going to go to the cytosol. Fatty acid breakdown in the electron transport chain happens in the mitochondria, and so it may be about compartmentalization. Okay, but we're going to talk now about glycogen synthesis. Okay, and the first thing is why, because actually we'll look at glycogen synthesis, and it actually costs energy to make glycogen from glucose, and you get some of it back, but you don't get all of it back. So why would the cell bother to make glycogen as opposed to just have glucose floating around? Yeah.
Isn't it more stable long term?
It's not actually about maybe more stable, but really what it's about is about osmotic pressure. Okay, so glycogen is made to store as a form to store glucose. To reduce osmotic pressure. So if we look at, this is an example of red blood cells, right? We can talk about isotonic or hypotonic or hypertonic, okay? Hyper, above, means that we have more ions outside the cell than we do inside the cell, right? I'm saying ions, but more, not ions necessarily, more solute molecules, okay? Because we're talking about glucose here. Is glucose an ion? No, it's neutral. Isotonic means we have the same number of solute molecules inside and outside, or the same concentration, at least I should say. Okay. And then hypotonic, which is what we're worried about here, is we're going to have more solutes inside the cell as compared to outside the cell. And so the problem, let's say we're talking about a liver cell. Liver is where a lot of your glucose gets stored. You also get your glucose stored in your muscles, all as glycogen. Okay, and if a liver cell, a relatively happy, full liver cell, had all its glycogen, all its glucose in the form of glucose instead of glycogen, it's got 0.4, so let's say it's a liver cell, which is a hepatocyte, would have 0.4 molar glucose if it wasn't stored as glycogen. What was the question?
So it would be twofold or something?
It would have lots and lots of ions in it. Okay, so what's going to have, not ions in it, lots and lots of solutes in it compared to outside. Because outside the cell, it's about, you guys remember normality?
Mm-hmm.
Normality is kind of how many solute molecules you have, right?
I've learned that, but I
Okay, okay. So your blood is most of the solutes in your blood is sodium chloride. Okay, and that's about 0.15 molar. So it's about 0.3 molar sodium and chloride ions, plus a little other stuff, some more stuff. But so we've got about 0.3 molar solutes in your blood. And you've got 0.4 molar glucose in here, plus whatever else you have, which probably includes that same amount of sodium chloride. So what that's going to cause is it's going to cause the water to want to come into your liver cells. And as a result, the liver cells would have to spend energy pumping it back out. And so they'd spend a lot more energy pumping water than the energy they put into making the glucose into glycogen. Does that make sense? Okay. So an average glycogen molecule contains about 50,000 glucose. So you can see that we're not doing the osmotic pressure equation here, but it's a lot of pressure. And so what you do is you then take 0.4 molar glucose divided by 50,000. And we're going to get down to a micromolar concentration of glucose here. So it looks like that's about 80 micromolar glucose. No, 8. I'd miss a 0. Okay. So much smaller concentration of, well, it's not actually glucose, it is glycogen. But you have a much smaller contribution to the osmotic pressure in the cell. So that's why this process happens. So let's talk about the process for the making of glycogen. What we're going to do is I'm going to use this diagram and I'm going to add on to the front of it here. But the step in terms of where you're adding on, you've got this UDP glucose that gets made. We'll talk about how it gets made. And the last step of adding something onto a glycogen molecule, a growing glycogen molecule, is glycogen synthase is going to take this UDP glucose and it's going to attach it to the end. So we've got this hydroxyl here, right? You could imagine we've got no phosphate there. So I think our attack is going to be, I'm not sure which way our attack happens, but we end up attacking here, making our attachment between these two glucose molecules, and we're going to release UDP glucose. You see that right there, or not, just UDP. So the UDP is acting as a leaving group here, right? It's a good leaving group. Okay, so this is the last step here. Later on in this course, we're going to talk about the regulation of glycogen synthase. So this is where we're going to have regulation here. We're also going to have regulation in the making of this, okay? Because once you have made UDP glucose, this is going to go into glycogen, all right? So the commitment step is going to be the step right before that. Why don't we go ahead and do that? That step involves UTP, okay? But we're actually not adding it just to straight glucose. We're actually going to add it to glucose 1-phosphate. So imagine this. Oh, I drew one of these in Gen Chem, and I couldn't use line angle. It just kills me when I have to do that. But so what we've got here is how many phosphates do I have going in between here and here? Yeah, 4, right? How many do I have in this? 2. And so out of that I get our buddy, pyrophosphate, right? You guys remember our friend pyrophosphate?
Wait, what is this process?
We're making glycogen, right? We're doing the beginning of making glycogen. Here, I'm going to drive you guys nuts by like putting my double bonds everywhere. Because it's a Friday before the weekend. Okay. And it doesn't matter. All right, so we've got our pyrophosphate we make there, right? Okay. But what happens to that pyrophosphate? It gets immediately broken down to make 2 phosphates, right? Here we see it again. You see this happens again and again and again. And how it's used is that this reaction right here, it has a standard free energy change of 19 kilojoules per mole, right? So this is spontaneous, and so it helps drag this reaction along. They're coupled, right? Think about it that way, or you just think about it as the concentration of this is super, super low because as soon as it's made, it gets broken down to make phosphates. But here we see it again helping us out. Okay, so we have glucose. Questions so far? Okay, the enzyme here is UDP glucose 1-phosphate uridyl transferase. I decided that was too long and I wasn't going to make you memorize it, so I'm not going to even write it down. Okay, but I do expect you to be able to draw this stuff, not this stuff down here, but draw this stuff out if given a figure. And part of it is because you already know this one, right? If you know the name of this, glucose 1-phosphate, you should be able to draw it because you already know how to draw glucose 6-phosphate. Just bring the phosphate over here. That's not a big deal. But where do you think we get the glucose 1-phosphate from? Glycolysis, right? Okay. And we're going to get it from our glucose 6-phosphate. It's glucose 1-phosphate. And this is phosphoglucomutase is what it's going to do. A mutase just moves atoms around. It doesn't take them away. It's going to move, often it'll be a phosphate, but it'll move some atoms from one spot to another on a molecule. So essentially, we're going to take it from glucose 6-phosphate. Questions? So this is from glycolysis, right? Second step, first step in the glycolysis, second intermediate. We're going to take it, we're going to make it into glucose 1-phosphate. We're going to add a UDP to it, and then it's going to definitely go on. It's going to be added to a one glycogen. Question?
Why is it like, why is uracil? Like, does that mean it's like an RNA?
Well, I don't know why uracil specifically, but it's a flag. It is a, when I say a flag, it's a tag. It's a, this glucose, goes on to glycogen. So it is a signal of that. And as such, we haven't talked much about commitment steps yet, but we are going to be talking about commitment steps. This is the commitment step. For glycogen synthesis. Which is just another way of me saying that once you do this, this thing is going on to, it's going to be added to glycogen, period. Yes?
Does, when we, like, if we have to write, like, anything, I know not necessarily specifically this, do you care if we draw the alpha or beta form, or if we just do a little squiggly line? Because in my notes, I've just been doing, like, squiggly both.
Actually, it should all be alpha.
Alpha?
Yeah. Because the only place we use beta really is like cellulose, right? You see, use beta actually in chitin, but that's not exactly glucose. But you're going to use beta when you're doing these structural molecules.
Okay.
You don't use beta really for anything that's going to be a glucose storage molecule.
What about like in the glycolysis and every other thing?
Those are probably all alphas. I haven't
Okay.
even looked at that. But I don't
Understand.
I don't really care so much about the stereochemistry. I don't know that I would grade on the stereochemistry anyway. Okay. What I'm trying to do, and the reason why you have to memorize these things is, I think I said this before, is that when I've taken other tests for MCAT, etc., that these things show up at this detail for glycolysis, and you'll see that we're going to lose detail as we go on, or you'll have to memorize less about these pathways because you're much less likely to see something after glycolysis. We might see something for fatty acid chemistry. We're not going to see anything for amino acid chemistry. So that's why the memorization of this course is the way it is. Okay, so once we made UDP glucose, now we're back here. And then this is destined to come down here and become glycogen. So what we do is we add more glycogen to the chain. Which end of the chain do we add it to? The reducing or the non-reducing end? The non-reducing end. Okay. The non-reducing end is this end. Okay. And so we add that. The reducing end's way over there. And it's the reducing end because it's the one that has the one carbon, which is a heavy acetal, which in the sloping can be an aldehyde, which can reduce metals, essentially, is where the naming comes from. Questions? Okay, like when last semester when we talked about glucose, or not glucose, glycogen, remember that glycogen was pretty highly branched? So we're not going to worry about making the branches, but I do want to show you this. So where glycogen starts is there's a protein in the middle that just acts as kind of an anchor. And then it'll start a chain, it'll branch off, and those branches will have branches, and the branches will have branches. And you keep branching, and you end up with this giant ball with 50,000-ish glucose residues in it. All right. And it has an important point with this is we have lots of non-reducing ends. So lots of non-reducing ends. I'm not pointing to all of them because I'm going to get bored, but it is all of them. And so if you think about building this up as opposed to building a big, long, straight chain polymer, I'm building this up, and once I have 2 ends, I can build it twice as fast. And once I have 4 ends, I can build it 4 times as fast, right? And so I can build this really fast, and then when I need the glucose, I can tear it down really fast because I have lots of ends to pluck things off of. Questions? All right, so that is glycogen synthesis. Let's talk about glycogen breakdown. It does not make it, it's a good question. Does it make it unstable? It's how fast it can break it down isn't whether it's stable or unstable. It's really about the thermodynamics, right? When we think about, well, it's not, it's a combination of the two, but enzymes are doing this. So we're going to talk about, later in the course, we'll talk about the signaling event for this. But if I'm low on blood glucose, do you know which hormone's in my blood? Glucagon. Okay. So insulin, if I just have a meal and I've got lots of glucose in my blood, insulin's released and it tells my cells to activate this process and build this up. But the enzymes are turned on. Okay. And then if I've got glucagon in my blood, all right, that means that I'm low on blood sugar and that I need more, that I need to break this down. My liver needs to break the glycogen down and release the glucose into my blood. And so those enzymes are activated. So it's not unstable because it's controlled by the enzymes, and it's a kinetic process. And both the reactions of building glycogen up and breaking it down are very spontaneous. So there's a very spontaneous reaction in there. In fact, let's look up here. Actually, let's look at the whole thing at once. Assuming I know what I'm doing, what reactions are very spontaneous here? This is very spontaneous, right? And we even talked about it being extra spontaneous with this. What else? This is also going to be very spontaneous, right? Okay. So that's also very spontaneous there, assuming that, you know, the authors of your book drew this correctly, and I think they did, right? Especially because you've got a UDP leaving, okay? How about this one? This is not very spontaneous, so it's not a commitment step. So your cells are kind of always taking, you could imagine that you've got this the phosphoglucomutase here. It's kind of always doing this. There's always some of your glucose 1-phosphate and your glucose 6-phosphate around, and if you get low on one, it'll drive over to the other due to the Chatelier's principle, right? So not very spontaneous. You've got lots of these. No energy is really used here to make this. And then when glycogen needs to be made, this gets depleted, and then more of it gets made through Le Chatelier's principle. Okay. All right, let's talk about glycogen breakdown then. All right, so as we talked about it, our non-reducing ends are where we're going to chew this stuff back. I'm not going to ask you to know. I'm not going to ask you to know the enzymes here. I'm going to pause because I've seen it on a test, but we'll just pass on this one. Okay. So we've got what you've got going on is glycogen. Once again, you've got this is your giant ball of glycogen. We'll call it glucose is your glycogen molecule. And you've got an enzyme glycogen phosphorylase that's going to nibble these things off. Okay, so it's going to nibble off glucose molecules. But look at this. It does not nibble them off as glucose. What does it nibble them off as? It's going to as glucose 1-phosphate. So instead of hydrolyzing them off, right? Remember, glycogen doesn't have phosphates in it. It's not like DNA or anything. There's not phosphates between those. So it's using a phosphate here, okay, for each one of those. It's going to use a phosphate to be its nucleophile and knock off these glucose molecules off that. Why is that useful? So this is spontaneous. I'll tell you it's spontaneous, right? Okay, all of these are spontaneous according to their arrows. I'm not sure whether, but that one's definitely spontaneous. Why knock it off with phosphate instead of not more state? It's not about stability. What was that? Okay, so we've seen them from previous. We've got phosphates around. We've got a lot of water around though too, right? Water is usually how we break bonds.
Put it right back into glycolysis.
You can put it right back into glycolysis, right? What goes on in our first step of glycolysis? It's not on here. There we go. Here's what happens in our first step of glycolysis? You add a phosphate. Where's that phosphate come from? An ATP, right? Okay, but if instead I could skip that step, and what I could do is I could use my phosphoglucomutase and go from glucose 1-phosphate. I just saved an ATP, right? It cost us energy to make the glycogen. We don't get it all back, but here we get at least a little bit of it back. Does that make sense? So we skip having to spend this. So glycogen, when you think about getting energy out of glycogen, when we think about our monomers, I'm not going to grade you on this detail, but we think about glycogen to glucose in our polymers, monomers. It's really when you go glycogen to glucose, it's really glycogen to glucose 6-phosphate, or glucose 1-phosphate, and then glucose 6-phosphate. I'll put the glycogen in here. Okay, so this is a way that evolution has decided to recapture some of that energy. And then what's a little interesting about this process, and I have seen on layer tests, is that we have this debranching enzyme. So essentially you nibble back these ends with your with the enzyme up above. We get down to where we have one of these branches, and then what you do is it's kind of funny. You take this bit with the branch, you move it to the end, and then you have a little bit, you pull off the one, and then you keep munching it back until you get into the next branch. There's a debranching enzyme, that's a special enzyme that's needed to deal with those structures. Questions? So maybe you know there's a debranching enzyme and it helps take care of the branching structure once you get too close to the end of the branch. I don't care that you really know exactly what happens there, but I do care that you've remembered or can relate this energy saving step where we're actually breaking a bond with phosphate as opposed to breaking a bond with water, which is usually how we break bonds in biochemistry, or in biology, really. Questions?
Can you write down the phosphate that you said?
Sure. So in the first step of glycogen breakdown, or well, it's not really the first step, it's the step, right? It's really a single step there. In glycogen breakdown, it's just separate from debranching. A phosphate is used to break the bond between a glucose and the rest of the glycogen chain. What do we call that bond? I didn't name it. It's a carbohydrate. What was that?
Alpha-1,4.
That's great. That's really specific. I like that, but I want the more general term. Glycosidic bond, right? Right. Remember, those are glycosidic bonds between the glucose and the rest of the chain. This produces glucose 1-phosphate. Actually, I'm going to make it more general. This produces a phosphorylated group glucose. And the first ATP using step of glycolysis can be skipped. Okay, this saves energy. So if you're going from glycogen and you take a glucose straight out of glycogen by this process, you've already paid for it previously making the glycogen, okay? You've more than paid for it. But if you take it out of this step, how many ATP would you get for glucose? Not 1, 3, right? You get 2 out, right, for glycolysis. And now you saved 1 at the beginning, so now you get 3. We'll do more counting. We'll do a lot of counting later, but that's just so we can think about how much we saved there. All right. I'm going to introduce this concept here. I don't think we're going to actually go over. There's a regulatory step here in glycolysis that we're going to spend a lot of time on. Okay, and the time we spend on it is going to be it's most of the regulation in this course. I'm going to have it's going to be reasoning problems, like you think about what's going on in the cell and what would make sense. This is the case where we have signal molecules, and we're not going to be doing that so much. Okay, it's going to be, it's actually probably outside this. It's outside the cell factors, like insulin's released in the blood, and it causes these things to happen. All right, but based on we only have about 5 minutes left. So what I want to talk about is this is the commitment step for glycolysis. There we go. This is the commitment step for glycolysis here. And what I mean is this step, right? Really, we're talking about glycolysis, this step, not this, even though they are sort of pointing at it. Let me go ahead and extend my arrow over. Okay. It's also a heavily regulated step for gluconeogenesis. All right, we have a set of compounds here that are going to turn this phosphofructokinase on and turn off fructose bisphosphatase, or vice versa. Excuse me. And in fact, the system we're going to go over is complicated enough that I'm going to have to number these. These are 1. Okay, so it's phosphofructokinase 1. You don't need to know about for your memorization task for glycolysis, but you will need to know it for when we talk about the regulation here. All right, and I did not make up the 1. Okay, it's the actual designation for these enzymes. Okay, so but we have this step, and this one is where most heavy regulation occurs. This also could be a good regulation step, right? Because we have single direction arrows. This one wouldn't be a good step because the arrows are double directional, right? And they use the same enzymes. You can't turn an enzyme on and turn an enzyme off. Why this step as opposed to this step? Why the third step as opposed to the first step? Yeah.
Skip past it, with G6P versus like once you get to
Right.
the third step, then you're just doing glycolysis.
Right. Once you get past, that's exactly it. Once you get past here, you're going down this way, right? But here we've got our branching paths. The things we make come off of there, right? So here we're going to go ahead. We could make glycogen. The way I've drawn my arrows there is actually, so we can make glycogen. We could do the pentose phosphate pathway, which is not reversible. Right? We have other things we make here. So we don't want the cell to go and regulate here because then we have to have this regulation step that's related to all this. We want the regulation for glycolysis to happen here after we get to the intermediate that could go off to other pathways. Does that make sense? Another thing that actually happens here that we'll talk about later is the liver cell will, well, actually, muscle cell. Let's talk about muscle cells. Muscle cells. What's the job of muscle cells? To contract. Right. So they're going to contract. So should they be making glucose for the rest of the body? No, it's not their job. That's the liver's job. All right. So what muscles do is muscle cells are designed that they take in glucose and they immediately make it into glucose 6-phosphate so it can't leave. Right. Your glucose transporters do not use energy to transport glucose in and out of the cell, so they kind of come in based on diffusion. But what the muscle does, it immediately makes it into glucose 6-phosphate. What does that do for diffusion? If we think about, I'm a liver, I'm a muscle cell. Okay, I'm a myocyte, and I've got glucose, and I need to make myself a bigger myocyte. I've got some reaction here where I've got glucose and I've got glucose 6-phosphate, right? if in the muscle cell, it's non-spec, I'm really driving this reaction. As soon as that glucose comes in, I make it into glucose 6-phosphate. What does that do with my concentration of glucose? It makes it really low. What is that? So how does that relate to glucose outside the cell versus inside the cell? It's going to make it want to come in, right? It's Le Chatelier's principle again, sort of. Does that make sense? Okay. So I kind of got a little off track there, but I also wasn't going to go into a whole lot of detail because you're going to forget in 5 days when we see each other again. But commitment step, this is where we're going to start next time. We're going to talk about this step in detail. I know it's Friday.